to one and all present over here to the third day of Professional Development Program on Innovations, Opportunities and Challenges in Life Sciences. Past 2 days have been very much exciting with a number of extraordinary speakers and their deliberation along with huge interaction between scientists. We look forward to a similar experience today as well. Now, I take immense pleasure in introducing and inviting another very promising and eminent speaker within us today. Our today's first speaker is Dr. Shilpi Minocha. She is presently working as a scientist at Kushuma School of Biological Sciences, Indian Institute of Technology, Delhi. After completion of her PhD from the lab of Professor Markus Noll in Institute of Molecular Life Sciences at the University of Zurich, Dr. Minocha served as postdoctoral fellow at Department of Fundamental Neurosciences and Center for Integrative Genomics, both in University of Lausanne. Currently, her lab at Indian Institute of Technology aims to understand the regulation of the molecular and cellular mechanisms that underlie the complex process of development and regeneration by using multiple animal model systems as well as organ models. To understand her working models and science underneath, I cordially invite Dr. Shilpi Minocha to deliver her lecture before us. Over to you, Dr. Minocha.
Thank you so much, Dr. Sumishta Das, for introducing me, and thanks a lot to MIT for inviting me for this talk. I'll share my slides. Are they visible? Oh.
Yes, Dr. Minocha, your slides are absolutely visible to us.
Okay, thank you. So I will start now. So good morning, all of you. Today I'll be talking about, some of the molecular and cellular mechanisms of development, regeneration, and disease that my lab in IIT is currently focusing on. But before I go on describing any of the results, I would like to begin with a short introduction. So slides didn't move. Did it move now?
No, it's not. Yes,
no,
it's now moving.
It has started moving.
Okay, I think it's a bit slow. Okay, so as you might already be aware, that during development, a human or any multicellular organism, for example, goes through a very amazing transformation. You can call it as dramatic, as a metamorphosis of a caterpillar turning into a butterfly. Now, over the course of hours, days, or months, the organism turns from a single cell zygote into a huge collection of cells, tissues, or organs. So, as an embryo basically develops, the cells of the body, they specialize into many functionally different types as the development goes on. Now, our bodies, or even that of a newborn, they contain a wide array of different cell types. It could be variable from neurons to liver cells to blood cells. And each of these cell types is basically found in only specific regions of the body. So the question we ask is, how is this intricate dance, basically unfolding? Now, this process of development is basically controlled patterns of gene expression, that are maintained during these multiple rounds of, cell division, differentiation, cell death that I spoke of. And then these gene expression patterns are actually controlled spatiotemporally. So it's actually a cohort of transcription factors, cofactors, chromatin regulators, which actually control these gene expression patterns. So my research interests have basically been vested in understanding, these complex mechanisms which underlie the process of development. I don't see the slides anymore.
Yes, Dr. Minocha, it is same with us also. We are not able to see your slides. Can you reload it once you
Yeah, I think the PowerPoint
to
crashed.
share the screen again.
Yes. I don't know what happened. Okay. Okay.
Yes.
So my questions, which I am interested in is basically how does these molecular and cellular mechanisms which underlie the complex process of development unfold. So my lab in IIT is actually using several organisms to understand this, but we primarily use zebrafish as a system to understand both development and also physiological organ regeneration, including that of the brain and liver. So in simple terms, basically what we aim to do is to identify reproducible patterns of cell behavior or tissue organization during these two processes, that is development and regeneration. And interestingly, what we have seen is that the mechanisms that control regeneration are both organ specific and also species specific. So we are seeing differences between, for example, between brain and liver, and also there are a lot of differences which exist between what happens in humans, mice, or fish. Now, what is also interesting for us to address is that none of these regenerative events actually ever lead to an oncogenic transformation. The tissue by itself knows when to start and also, most importantly, when to stop. So how is this balance between regeneration and cancerous transformation maintained? So zebrafish basically, is a very good model for studying this because of its genetic tractability, its optical translucency. We are able to do a really high order microscopic live imagings, and also it has a very great deal of physiological similarity to humans, which makes it well suited for us to to study these processes of development, regeneration, and also a wide range of diseases, which can of course lead to advances of drug modeling, discovery, and also potential therapeutic strategies. So because of limitation of the time, I'm only going to speak about the first aspect, which is brain development and regeneration today. So in zebrafish, basically, it's a very widely used model organism for neuroscientific studies. It has many common features to vertebrate brain structures, together with very, very similar neuroanatomy and also neurochemical pathways, which are directly relevant to human diseases. Now, however, the brain of the adult zebrafish is a bit different to that of the mammals, that it has a very intense neurogenic activity. What I mean by that is that the zebrafish brain is capable of making newer cells even when when it's an adult fish, and this intense neurogenic activity is basically the baseline of its regenerative properties, something which I will be broadly speaking about today. So what we do is basically we try to mimic brain injury. So what's brain injury? So brain injury in very, very simple terms is when a bump, a blow, a jolt, or any kind of head injury causes damage to the brain. Now, this is widely called as traumatic brain injury. Now, traumatic brain injury is a leading cause of death and disability. It's an international public health concern. Every year, nearly 60 million individuals worldwide, sustain a brain injury. And out of it, several of them, up to 2% of these people, they actually survive, but with neurological disabilities caused due to this brain injuries. Now, like I said, the brain injury is basically when an external mechanical force is going to cause a disruption, and then this can also alter the brain functioning. So now, although, I'm using the word brain injury, it is discussed as a single clinical entity, but in fact, it's a complex and heterogeneous pathology. And there are a lot of cellular and molecular events which actually happen directly, immediately, or a little while later post brain injury, and which are right now a goal of a very important preclinical research and spurring, expansion of a lot of different mechanisms. Now, as I said, the incidence it's very widespread. It's not something which only happens in one area or the other. This is something, the casualties are widespread. And then, as you can see here, this is showing, the causes of damage. It could be as, sparse from like automotive collisions, could be a fall, could be a blunt impact, like here in a head injury. It could be any kind of an insult, basically, which can lead to traumatic brain injury. Now, brain injuries, they can be classified into different forms. So, for example, depends, what caused the injury, what is the severity, how much portion of the brain was affected, and other parameters. So broadly, if I speak, it's divided into two mechanisms, which is either primary injury or secondary injury. Now, primary injury is something which happens, immediately after the impact. The secondary injury is more initiated, although initiated at the moment of the impact, but is believed to continue for many years through, a series of cellular, physiological, and molecular processes which affect all different kinds of cells, like, likes of which some of them are mentioned here. Now you can say that the primary injury basically is the one which causes the greater damage to the brain. And the secondary injury is the vital role which is happening, once the person has survived. And, other factors like inflammation, edema, ischemia, affect which accumulate over the period of time and can lead to causing some kind of neurological deficits. Okay, now the injury, when it is caused by any kind of trauma or a stroke, it sets a lot of things, a lot of emotions into practice. The brain basically, has certain kind of combat mechanisms. It will try to repair, it will try to circumvent the damage. So how does it do it? So the mammalian brain, our brain basically, has very broadly speaking, two mechanisms. One, either there can be adult neurogenesis. What that means is basically, the brain is going to try to replace the damaged cells. Now, whether or not this neurogenesis basically takes place in an adult human is actually very controversial. Some studies have even failed to demonstrate any kind of proliferating cells in the adult, central nervous systems, whereas others, they have demonstrated the presence of equally, convincing fashion, some of the dividing cells. Now, so whatever its role in the adult, basically, the neurogenesis, as the word, of course, as by itself suggests, it has the potential to repair the injury if it can be induced in some way. So it is believed that there are certain kind of quiescent stem cells that exist in the brain, which can be, which come into action once there's a phase of injury is there. What is also known is that this process of neurogenesis it diminishes with aging. So at cellular level, the brain is also a bit plastic. That is, it tries to recover. So it could be different mechanisms. For example, it could try to remap the neural circuits, whatever damaged cells are there in the brain. There could be newer processes, could be formed. There could be more outgrowths. The pre-existing cells may try to make more connections, Basically, the aim is to limit the damage as much as possible, whatever damage has happened, to contain it at a particular place so that it does not go ahead and then, to somehow come back to the state, the homeostatic state. Now, in several model systems, people have tried to express some kind of growth factors, neurotropic factors, which can be increased after injury to accelerate these processes of neurogenesis and brain plasticity. But the problem in the human brain is that, in general, the human brain is very hostile to these processes. It actually exhibits inhibitors of axon regeneration. It is kind of stopping the whole thing from happening. There are a lot of inhibitors happening present over there. So if you look at the human brain in this schematic, you can see that these are basically the two regions which are known. So these are called as the sites of neurogenesis in the brain. And that is where the new neurons are formed. So one of them is this hippocampus, as is shown here. So this area, which is shown here in the middle, it's the dentate gyrus. It's a specific area where neurogenesis can happen. And the other zone is this subventricular zone, labeled here, which is also shown in blue. So one way, to detect any kind of newly formed cells is, to do BRDU staining, which you might have heard. So BRDU becomes part of the DNA of the new cell and can be seen under a microscope. So this picture, if you see over here, so the green ones are basically the neurons, which are the older neurons, and it's showing this little area of the hippocampus. Whereas if you can see, these little red cells over here, these are the ones which are the newer neurons, which have BRDU in their DNA. So, so these are the only two regions which have the capacity to generate newer neurons in case of any kind of injury happens. But the other part of the brain do not, or there has been no report as such that they can generate any kind of neurons. So now this becomes a big problem. So, there are only small two regions. So if you compare now this human brain to that of fish brain, so this is the human brain on the top. It's actually showing, a side view. So if one cuts, let's say from the nose to the back, and, you're cutting starting from the ear, this is the view that you're going to see. So this red area is basically the only area which is capable of making new neurons. And if you compare that to a zebrafish brain, which is here below, so you can see that these red areas, they're all along the entire brain axis on the side. So there are only two areas which can make new neurons in humans, whereas there are nearly 16 neurogenic niches in the zebrafish brain, which can actually make newer neurons. So because of this, privileged location, if I may say, of these neural stem cells in this zebrafish brain, and this exceptional regenerative potential of this brain, it makes the zebrafish basically as an attractive system to understand and to understand the processes of regeneration in general. So if we now go to the next slide, wherein you can see the neural stem cells. So here we are comparing zebrafish, in the middle is rodents, which is mice, and also human. And just a section through all of them, just comparing, what I've already shown in the last slide. So you see here the neurogenic niches where new neurons are formed. This is the same region which you see here in mice or in humans. So you can see here the different mechanisms. So it looks pretty much the same. However, the regenerative potential of zebrafish is many-fold higher compared to that of the mice or humans. Can you still hear me?
Yes, you are absolutely audible.
Okay, because we have lost power here. Okay, so anyway, so you can see here in panel D, it's showing actually the regenerative, the cells. So this VZ is actually the ventricular zone. So these are the stem cells here which are lining these little areas. So these are those which actually give rise to the newer cells. Now, all these regions over here, they have the capacity to generate new neurons. So, however, in contrast, to these two places, which is in mice or in humans, what is new in zebrafish is basically what happens is that when the new neurons are formed from these stem cells, they are directly deposited, just under these stem cells. They do not migrate normally. And if there is an injury, then only they are going to migrate very longer distances and they get intermingled with other cells. Now, the traumatic injury basically enfolds, a very specific program in these neural stem cells and these progenitors to make these new neurons during regeneration. So here, again, I'm showing you the zebrafish brain, and these green ones are these neurogenic niches where they can make new neurons. In B, you have a higher magnification of this schematic showing the neurogenic niche again. So these are the progenitor zones, basically the green cells, and also these blue ones are the intermediate progenitor cells, and all these ones in the brown are those which are the new neurons which have been formed. And basically this is what happens when there is any kind of injury in the zebrafish brain. These stem cells, they kick into action, and then they're capable of making these new neurons, which will then migrate to larger distances to populate the damaged brains. Okay, so whether this causes any kind of restorative neurogenesis is basically something which has been discovered in the last 2 decades, and now scientists are actually trying to understand the different molecular and cellular mechanisms which unfold this restorative neurogenesis mechanism. So if we look a little bit more into the next slide here, you can see one of the brain hemispheres. So imagine that there is an actually injury here in the middle. Now I showed you that these neural stem cells, they are here in the periphery. So the brain then is capable of actually recognizing that some injury has happened, and this kicks these cells, which are there in the neurogenic niches, into action. This has happened, this is happening by several cells. So they're like microglia, oligodendroglia, and some other radial stem cells which start proliferating to make newer neurons and newer glia, which then kick into action. So the newer neurons here are shown by this, the orange mark, that is a newborn neuron. So then here what is happening is basically the proliferation is peak in this region, which is the ventricular zone. And then this is going to make newer neurons. And then they are going to get accumulated here in the subventricular zone, and then they are going to start, migrating. Now, most interestingly, what happens is that never, any of these events, they never lead into any kind of oncogenic transformation. It kind of uses, so there could be different pathways that this process is going to actually use. So it is either, there could be activation of already something which was pre-existing, some developmental pathway, just having a new function now. Or there could be some pathways, which are having similar function as, is in the case here, that they were actually generating neurons, but then some of them are deactivated. Then there is activation of some new or injury-specific signaling pathways. And lastly, there are some which are actually, which are actually injury-specific, but they're promoting the activation of these neural stem cells. So there are already distinct markers of neuron and glia, which I will also be showing in this study today, which already exist. So it's possible to actually look at stem cell populations and also these newly generated neurons and glia in these brains. So a lot, of course, is known already during brain regeneration. So it's already known, what kind of cells produce these new cells, how are they integrated, and it's happening without a scar formation. Glial scar is something which is formed in human brains, and then different pathways people are now trying to look into. But there are several aspects still which are unknown about brain regeneration. So there are still some population of cells, it's not known that how do they migrate, how do they actually integrate these new neurons into pre-existing circuitry. There's a very extensive genomic duplication in zebrafish, so it's not known whether what kind of tuning of these programs happen, and also a lot still is not known about the cellular molecular mechanisms. So in our lab, we basically try to mimic this process of brain injury. So you can see here, so we take fish to generate this telencephalic stab wound injury model, and we put them into Tricane. It's an FDA-approved drug, which is basically to anesthetize them. And after that, we take the fish and we poke with an insulin needle into one of the hemispheres of the fish, and the other hemisphere serves as a control. So, and then we look at different aspects. So these are the 3 students who are actually working on this project in my lab. And here you can see that, it's the same thing. We actually do an injury to one of the hemispheres of the brain. So this area is called as the telencephalon. You can see here that the injury is clearly visible. And this is one of the sections, which is a histological section through a paraffin slide, where you can see again the injury here in one of the telencephalic hemispheres, where the other hemisphere serves as a control. And if we do any kind of BRDU incorporation or look with any mitotic marker like PCNA, we see an upregulation only at the poke site. That is where we had the injury, which is also clearly visible through this Western blotting. So after this, our key objectives in this study is to look at gene expression analysis to find common gene pairs between both regeneration and cancer studies. So we compare our transcriptomic data obtained from these fish to that from the low-grade gliomas from patients from publicly available repositories. And we are also interested in understanding the different molecular and cellular pathways which in general exist, this regenerative process. We are also interested in understanding the signaling pathways with special focus right now in the lab is on notch signaling pathway and hippo-YAP signaling pathway. And lastly, we are also interested in interspecies comparison and also inter-organ comparisons between these for this process of degeneration per se. Now, before I go on to describing some of the results, I would like to introduce you to a molecular factor which we identified to be relevant for our studies. It's called as host cell factor 1 or HCF1. So host cell factor 1 is basically a very large 2035 amino acid protein with different domains. And the one shown over here in green is the Kelch domain, which is known for most of its interaction. So it's basically a transcriptional co-regulator which links histone negative and positive histone modifying activities. It gets cleaved at these 6 centrally located proteolytic repeats through an enzyme called as OGT. I will be introducing that later. So HCF1 has been known to play a very important role in cell proliferation. And through my studies in the recent past, we have found that HCF1 is very important for liver regeneration studies in mouse livers. So for nearly 2 decades, HCF1 was only known as a cell proliferation factor. But in the last decade, newer studies have actually connected HCF1 to several other roles such as metabolism, regeneration, and also mental retardation. It's a highly conserved metazone transcriptional co-regulator, as you can see here. So here it's a comparison between human, mice, fish, fruit flies, worms, planaria, anemone, and also sponges. So you can see here that in all of them, there is this presence of this Kelch domain, the one which is primarily the interactive domain with other proteins. The proteolytic repeats are present only in human, mice, and fish. So the size of the protein is a bit different between the human, mice, and fish. And in fish, there are actually 2 orthologs of HCF1, that is HCF1A and B. And what we have also seen is that actually HCF1 plays very variable roles across different species. Now, I will not explain all of them, but what is of relevance today is what we found with the brain study. So in fish, basically, it has already been seen in the past by other groups that mutations in HCF1A and B, both the orthologs, they led to increased number of neural precursor cells. And then there was some kind of cranial defects which were observed. Whereas in mice, mostly the studies have actually shown that the loss of HCF1 either led to increased cell proliferation in hippocampal neurons which were taken into culture, or there was actually no effect on cell proliferation at all when I saw them in the brain. And on the other hand, in humans, HCF1 has been linked with a lot of excellent intellectual disability patients because HCF1 is actually on the X chromosome in both mice and humans. And as you can see here, that there's a little bit difference between the amino acid, the number of amino acids, the number of exons, and also the proteolytic site, which exists between zebrafish, mice, and human. So I will actually begin by explaining what I have seen about the role of HCF1 in mice, and then I will come back to the zebrafish brain. So first of all, just a second, yeah. So what I've seen is, so for example, this is a very early E7.5 mouse brain. You can see here the wild type. This is a bright field image. So this portion over here in the below is actually the epiblast, which is the embryo proper in these early mice. And then this is an HE staining of a section. You can see that HCF1 is basically expressed throughout the embryo and also in the extra embryonic parts. So I did a knockout of HCF1. And what was found that this embryonic part was much smaller. There was complete loss of HCF1. And these embryos, they did not survive, so they died due to a defect in gastrulation. So basically, the loss of HCF1 is lethal for these embryos, the mouse embryos. And what we saw, that when we did the labeling in the mouse brain, so this is a labeling with beta tubulin 3, which is a marker for neurons, which you can see here. This is actually in the hippocampus. And if we overlay, this is with Hoax, it's actually a DNA, it's a nuclear stain. And then we did overlay with HCF1 antibody. So if I remove all of them, you can see that actually primarily HCF1 is primarily expressed in it's nuclear in these neurons. On the other hand, in mouse glia, that is in the astroglia, the other type of cells, it looks like that HCF1 is not only nuclear, but it's also cytoplasmic. So here is a stain of HCF1 in red and Hoaxed, a nuclear stain in blue. If I remove the blue, you can see that it's also expressed in the cytoplasmic processes, while it's also there in the nucleus. So if I now overlay these cells with actually a glial marker, which is GFAP, you can say that there is a full co-localization between these processes, which is which is making it both nuclear and cytoplasmic in the astroglia. So first aim that we wanted to do was to generate a conditional knockout, because as I've shown to you two slides earlier, that HCF1 knockout is actually lethal. So what we did was we actually tried to remove HCF1 through either both neurons or glia, and we used different strains. I will only present the results for one of the lines, which is NKX2.1 Cre. So NKX2.1 Cre is basically labeling a subset of neurons and also glia in the brain. And when we actually removed HCF1 in both neurons and glia in the brain, what we saw was that, so for example, in the MGE, this is a medial ganglionic eminence. It's a region in the embryo which is responsible for making neurons and glia during brain development. So this is an early embryonic brain 12 days after birth. So if you can just see here in the green one, so GFP is basically labeling all the cells which are NKX2.1 derived. This is a wild type brain in A2, and you can see that there are some cells which come out of this progenitor zone and they start migrating to occupy higher order areas. But if you compare A2 now to B2, which is actually a HCF1 knockout, you can see that the cells which are exiting this medial ganglionic eminence of progenitor zone, they are largely reduced in the HCF1 knockout brain. So it may appear here that maybe the HCF1 actually cells, they are not migrating properly. But we also saw that upon labeling with the cell proliferation marker Ki67, that there was no effect on the proliferation. This ventricular zone is the one which has the progenitor. On quantification, we realized that actually there was no effect of the proliferation of the progenitor cells. So it probably, they are generated, but something happens to them later. So for that, we did labelings with a tunnel. So this labels all the apoptotic cells. So if you see here in A2 and C2, so these are actually showing, the tunnel apoptotic cells in wild type embryos. And here is E14.5, which is 2 days after E12.5. Again, in wild type, you can see some of the apoptotic cells. And if you just compare here on the right-hand side, in B2 and D2, you can already see that E12.5, there's a significant increase in the number of cells which are actually apoptotic. And this number is also more increased in E14.5. So what it comes to that, although the progenitors, they are replicating, maybe the neurons in glia, they're still getting generated, but once they're generated, they're not able to migrate. And secondly, that there's increased incidence of cell death in these two populations. Now, if we actually cut the, this embryo, so this is E18.5 embryo of the mouse, and if you cut it in this way, which is a coronal section, and if we look at the 0, which is in the middle, so this is how you actually see a section. So you see the two hemispheres, and they are linked here in the middle. This is called as commissure. So basically, this is the one which is linking the two brain hemispheres. And here you can see another commissure. It's called as the anterior commissure. So this is the view I will be showing you in the upcoming slides. So the idea for us was to actually look at the entire brain and see whether the loss of these neurons and glia, which I've showed you in the last slide, is it leading to any kind of phenotypic alterations or not. So in general, what we saw was this is a label, this is an NCLEX 2.1 tree reporter line where, like the last slide, the GFP is labeling all the NCLEX 2.1 tree. So you can see here, this is the wild type showing different regions of the wild type brain and labeled with GFP and DAPI. And if you see here in the HCF1 knockout brain, there's a marked reduction in the number of neurons in glia, for example, which are present in the cortex. So this region is called as the cortex. So you can see here also, and this is the commissure here. So you can see here also in the commissure, that looks like that there are less number of cells. And this one, which is the anterior commissure here, it appears that, something is wrong here. Maybe it's not formed properly. So if we have actually a more closer look to these regions, you can see that, for example, here in the corpus callosum, so this is the corpus callosum, the commissure, which I showed you, which is linking the two brain hemispheres. There is a reduction in astroglia. So this is GFAP, an astroglial marker. So you can see that there's less green in general. And also, this corpus callosum looks a little bit thinner and is also not formed properly. So in general, there's a reduction of these glia. So from these progenitor zones, which I showed you, medial ganglionic eminence, the neurons, when they are, or glia, when they are formed, they go and migrate to these other places. And in general, it looks like that, lesser number actually reach this commissure. On the other hand, I showed you this other commissure, which is the anterior commissure. This is labeling L1. It just labels all the exons, basically, which pass through these commissures. So you can see here in the wild type, in red, this is how the anterior commissure look. There's a thick bundle of callosal exons, which passes. And this is a labeling of GFAP, which is astroglial cells, which are actually present in this anterior commissure. So now if you compare this on the right-hand side to these HCF1 knockout mice, you can see that just very grossly that these exons which are passing, they don't look normal. They look a bit haphazard. Instead of all passing through, in the middle as a tangential migration, they're going up and down. And not only that, but if you also look at these glial cells which are present, they are more haphazard. They're no longer arranged in this way. Instead of being horizontal here, they look more like vertical or, going in all different directions. So lesser number of cells have managed to reach these commissures, and those that have reached, they are highly disoriented. So there is a major problem with, migration of these cells and their survival upon loss of HCF1. Now, if you look here again, these are the sections which you can see, this is a normal stain. It's a Nissl stain, which just labels all the nuclei in the brain. So on the left are, different brain sections from the wild type. So this is just, it depends, if you imagine you're going from the nose to the back of your head, this is how, the different sections are going to look. So if you look, the cortex does not look very normal here. So the cortex actually, this is called as a unilateral polymicrogyria. So basically, if you have ever seen, any kind of schematic of a human brain, you might, there are a lot of gyrations, it's a multiple foldings which happen. So usually those foldings are actually absent in a mouse brain. But if you see here upon loss of HCF1, we have these gyrations which happen. So this is a higher magnification picture. So if you compare on the wild type, which is G and I on the left, to the knockout brains on the right, you can see here that, they're indents, they are folded. And also these cortical, layers, they look a little bit more squished. So this is a model of polymicrogyria. So this is a wild type. So all the cortices in the brains, they are actually divided into 6 layers. So these layers are 1 to 6. So this is actually from this image has been taken from a paper from another study where they had actually identified polymicrogyria. And if you look here on the right, which is actually the mutant, which is the HCF1 knockout brain, it resembles this and this structure, and the layers are somehow squished. So I went about, and there are distinct markers for all these layers. So layer 1, 2, 3, 4, 5, 6, they're all having some kind of markers. So I used all those markers to basically look at what is happening. So I will just point to what is, what is significant. So we saw with relin, it's a marker for layer 1. So compare A1 to B1. So there was a reduction in layer 1. There was also a reduction in other layers, but it was not very, very drastic. So when we looked at, in the next slide, you can see TPR1 is another marker. It's actually for layer 6. The last layer, it was actually, if you compare O1 to P1, it was markedly reduced. So if you just give a very quick glance to the other lines, you can see that here, for example, CTIP2, which is a marker for the layer 5, it's also reduced and it looks like it's more narrower. Similarly, cal binding, which is actually ranging from 2 to 5, it's also looking that it's a bit more reduced. So in general, the different layers in the absence of HCF1 are actually reduced. So in general, the loss of these neurons and glia in the mouse brain, they lead to their increased depth. It leads to these problems in the cortex. You saw these waves in the cortex, the problem with the commissures. And also, in general, there is not only that the cortex is this different gyration patterns, but also there is a problem in how the layers are formed. So basically, this work so far tells that HCF1 has some role to play basically in the mouse brain. Now I'm actually going to make a transition from here and link these results to the zebrafish brain. There was actually there was no mouse facility. There's no mouse facility actually in IIT. So most of this work has been done in collaboration with my past lab in Switzerland, while I was there, because I could bring a lot of samples back. And also, because of my project with another lab in INSAM, I have a CIPIFRA project with them. So they house the mice and they send us sections here, or, my student goes there and brings the things back. So I actually set up a zebrafish facility here in IIT. And all these results, basically are all post-COVID. So you see here, this is HCF1 and zebrafish brain. So we were lucky that actually the HCF1 antibody that we have is also recognizing both the orthologs in the zebrafish brain. We have looked at the knockdown system also, and we have actually already confirmed that, the antibody is recognizing specifically HCF1. So here in the middle, you can see zebrafish compared to mouse brain and also human cell line. So these are the different proteolytic fragments. And, the proteolysis is a bit different, but it's expressed throughout. And there is no preference as such to any of the orthologs in the zebrafish brain. Both HCF1, HCF1A and B, they're both expressed in zebrafish. So we began actually first by looking, similar to what I did in mice, is that what happens if we lose HCF1 in fish. So, we did this morpholino-mediated knockdown of both HCF1A and HCF1B. And this microinjections were done, as soon as the embryos are collected at 0 hours post fertilization. So we do the survival studies a day later, and then we do the imaging 5 days later. So there was a reduction in the survival of these HCF1 morpholinos, both A and B, but they did manage to survive, nearly 50% of them. And those that survived, you can see over here in HCF1A and also in B, they look a bit stunted. The HCF1B has more marked defects compared to HCF1A. So both of them are shorter, thinner, and there is some kind of edema that happens here, which is an heart edema. And also here in HCF1B, they look a bit more curved. So we followed them, and then they managed to survive. The HCF1A are surviving much better than HCF1B. The HCF1B, they die around 9th day. So HCF1 looks like it's very important for regeneration, for development. And then, it was now for us to see whether it has playing any role in regeneration, because as I said, in one of my introductory slides, it's very common for actually the system to reutilize its resources. So something which actually plays a role during development, it's been seen that, the same factors there again actually, used during also regeneration or any kind of injury specific mechanisms. So we saw that actually when, there is injury in the zebrafish brain, so this is a control showing HCF1 proteolytic fragments. And if you compare that to 4 days after injury, 7 days, 14 days, or 30 days, we saw a marked increase in the presence of HCF1 in the regenerating zebrafish brains. And here on the right, you can see that, the left is actually basically serving as the non-poked or the non-injury hemisphere. And on the right-hand side is the hemisphere we do the injury. And you can see that, the circle region is the one which is showing that there is actually the injury spot. And there is a marked increase in the localization of HCF1 positive cells. So not only, there was a marked increase in these HCF1 positive cells, but also when, we look at the co-localization with the proliferation marker, which is BRDU. So I'm showing the regenerating hemisphere here, and different areas have been marked ABC, wherein we see the BRDU positive says immediately 4 days after the lesion. So it's really early time point after the injury has been done, and these ABC, they're shown actually also in enlarged here. So you can see that all the HCF1 positive cells, they're co-localizing with BRDU positive proliferating cells in all these different enlarged regions shown in yellow, by the merge. Now, this is 4th day, which is very early after regeneration. We see even a higher robust response happening, let's say 7 days after regeneration. So you can see here that the number, just a second, sorry. So here you can see again different regions. This is the injury spot, and all of them actually have again HCF1 co-localizing with HCF1 with BRDU positive cells in all these different A and B regions. So once we know that they are actually labeled in all the different proliferative zones, the first thing we wanted to see was whether HCF1 was also upregulated in the stem cell population. So for stem cell, there's a marker that is GFAP. It serves, and you can see here, this is the ventricular zone in the middle. And red is actually all these stem cells. And then when we looked at, this is a merged image. This is 4 days after very immediately response in A and B. You can see that HCF1 is directly co-localizing and is heavily expressed in these progenitor zones already, and also not only in the progenitor zone but also in the newly synthesized cells. So this is 4 day. This is a 7-day brain where you can see, and already by the red, that the response is now robust. These cells this marker is not only labeling the stem cells, but also newly generated glial cells. So you can see here A is a labeling in the ventricular zone. So there is a labeling of HCF1 with GFAP, and B is basically a staining which is happening in the the newly generated glial cells. Now this GFAP is basically a cytoplasmic marker, and HCF1 is primarily nuclear. So we have actually looked at the co-localization also with DAPI in this area, and we could not find any cell which was not HCF1 positive actually. Now the regeneration process is primarily largely over by 14 days. So you will see that here in 14 day, the ventricular zone is actually now less labeled, and it's more the newer cells are now more towards the center and in both. So A is showing a central region where the newer cells are migrated, and B is showing a cell which is the ventricular zone. So you can see that HCF1 is present in both these areas. So we are convinced that HCF1 is upregulated in the proliferating zone, and is also present in the stem cell area. So we checked this in the glial population. The next obvious thing for us was to also check in the neuronal population. So there are different markers. I'll only show 2. So we have checked for neuro D1. So we could see that there was an increase of HCF1 labeling in the the newly generated neuronal cells, and you can see here below also another area. It's a section, confocal section, to show a better clarity. And here it's HUCD is another marker which is labeling the newly born neurons. And again, we could see that here you will see through one section of the confocal image that it's highly nuclear and also expressed in all these different areas. So HCF1 is basically upregulated in these newly generated neurons. So now we wanted to look further. So I told you in my introductory slide that HCF1 is a big protein, right? And for its proteolytic maturation, basically what happens is it's another enzyme, OGT, which cleaves it, at its centrally located proteolytic repeats. And then, it's actually at the N and the C terminal of HCF1 do different functions. So this enzyme, it's OGT, or glutamate transferase, which does it. So we saw that like HCF1, OGT is also upregulated during brain regeneration in a similar fashion. So if you see here compared to control, in 4 day, 7 day, and also 14 day, there's a marked increase in the presence of OGT. So now OGT, as I told you, so OGT basically, it's an enzyme which is actually doing this addition of these as you can see here, it's actually doing an addition of these N-acetyl glycosamine to serine and threonine residues, of different nuclear and cytoplasmic proteins. So this modification is done by OGT, and it's removed, these O-glutamate modifications, they are removed by cellular glycosidase OGA. So you can see here on the below, we also checked for OGA, but we did not see any increase in the presence of OGA, the one, which removes. So it's primarily the increase is there in OGT. And then we use actually an inhibitor, OSMI. So OSMI1 is an inhibitor for OGT action. And then we tried to compare the levels. So you can see here in control versus, during regeneration or DMSO control, and, when this drug is maintained, there was increase in OGT levels, but when we use OSMI, there's a decrease in the OGT levels. So it means that, the inhibitor is working well in a zebrafish model system. So then we wanted to see what would be the impact of the loss of OGT here. So if you look here in these slides, upon administration of OGT, so this is a normal 4-day post-injury brain, and you can see here there is an increase in HCF1 at the poke site, and there is also an increase in PCNA, which is an S-phase proliferation marker. In these brains, which are treated with OSMI1, which is an OGT inhibitor, there is no increase as such in HCF1 that can be seen in the poked hemisphere, and also there is very little PCNA which is there. So this is also visible in this Western blot, which you can see here. So during brain regeneration, PCNA is increased, whereas upon treatment of OSMI1, it looks like clearly the PCNA is not increased. So it looks like these brains, they fail to regenerate properly upon loss of OGT. Now, the other obvious thing for us to check was, like I said, OGT is causing the attachment of these N-acetyl glucosamine residues, so glucnac moieties. So there's an antibody called as RL2, which is capable of recognizing these O-glucnac moieties. So we checked also by immunostaining and immunoblotting. So you can see here in immunostaining, there's an increase of HCF1 at the poke site. It's not there in OSMI like in the last slide. There's an increase in RL2 staining in these poked hemisphere, but it cannot be seen in these other hemisphere, which is treated with OSMI. Similar results could actually also be seen with the Western blot, that, you can see there's a reduction in RL2 and HCF1 levels upon OSMI treatment. So therefore, clearly it looks like, that HCF1 OGT axis both are actually having a role to play in regeneration of the brain, and it's impacting the glycosylation. Now we are in the process of investigating the effect, and I'm actually doing this in collaboration with Dr. Vaibhav Kapuria at MIT. Now, after we saw these brain region effects, so now the problem is that, in the adult, we cannot target HCF1. Like I told you, that these morpholinos injected knockdown HCF1 embryos, they're dying. So we cannot use them for our brain regeneration studies. So one, of course, a way was, to use the inhibitor of the OGT, which is actually the contracting pattern of HCF1. So we decided that, we have to do something and also look at, in general, about what happens to HCF1. And so for that, we actually resorted to using another region, which is retina. So retina is, is another part of the CNS, right? Like the brain, both retina and brain, they have similar mechanisms, have been shown in mice for their development, and they are both part of the central nervous system. So in collaboration with the lab of Dr. Rajesh Ramachandran and Isar Mohali, we did morpholino injections of HCF1A, B, and also both of them together in the adult retina. And then we looked at EDO. So, basically this is a chemical which is looking at, the proliferating cells, and we harvested these retina on day 4. So, so here, I will guide you through these cells. So you see here, this is control. That's how the proliferative zone looks in a control retina, which is actually after 4 days of injury. So basically, typically one should be able to see, these proliferating cells. Lissamin, so these morpholinos, they're actually tagged to a fluorescent probe, lissamin tag, so that, we can see whether our morpholino injections have worked. So you can see that, the morpholino injections had worked. And here we just use a control lissamin as a control for the fluorescent probe labeling. And here, and normally you can see that there's a lot of proliferation happening in all these layers. But upon, after 4 days injury of those embryos which had HCF1A, B, or both double knockout, there was actually a reduced labeling of EDU in A, B, and more so in when both of them were injected together. So it means that HCF1 is actually playing a role also in the retinal regeneration, that, upon loss of both or either of them, there is actually a reduction in, in the proliferation that is happening in the retina. Now, in the next slide, you can see here that actually this is the quantification which we did. Sorry, just a second. So this is a quantification we did, so you can see here that the labeling of HCF1A, B, it's reduced quite a bit, and also in double knockout, it's more reduced. So something also, very unique we found was that, we were checking for our antibody. Let's say this HCF1, you can see here, the HCF1 antibody basically can recognize both the orthologs. So compared to the control, you can see there's a reduction in HCF1A and HCF1B, and there's a marked reduction when we remove both. There's a knockdown. So the antibody, this is a test also for antibody and also for the knockdown, both working. So we actually tried for also other things of interest. And then, we came across this protein called as YAP. So we saw that upon loss of both HCF1A and B, there was actually a reduction in the levels of YAP protein. So now what is this YAP protein? So before I go on to describe that, so this YAP protein has been shown actually by study by another group, Peng et al. In a paper in 2017, that YAP actually interacts with OGT. And you all have seen, through the past slides, that HCF1 is actually also an interacting partner of OGT, because OGT is required for its proteolytic maturation. So it looks like, there might be actually a connection between HCF1, OGT, and YAP. So we were very enthralled by this and we wanted to check more. So now this YAP protein, I will explain more about it. It's actually an integral component of a hippo-YAP signaling pathway. So we checked, we were very curious, okay, that YAP is going down. So we went about checking for other components. So these are all different downstream regulators of, downstream effector moieties of this YAP protein. And we saw that in general and retinal regeneration, all of them, several of them, they're actually part of hippo-YAP pathway. They're all going down. So the red one is HCF1A only, B, and HCF1 plus B morpholino injection. So you can see that the levels through qPCR, that the mRNA of all of them were going down. So clearly it looks like that upon HCF1 is doing something to this particular pathway. And perhaps maybe it is doing it through YAP, because YAP is the one which is controlling the levels of all these transcriptional targets that I've shown you here. So these are all basically YAP transcriptional targets. So what is YAP? So YAP is basically final effector of a hippo pathway. It was first discovered in Drosophila melanogaster. It's a very key pathway which is known to maintain organ control and homeostasis, and it's highly conserved. So when hippo is on, YAP is basically sequestered in the cytoplasm and is proteolytically degraded. And when this pathway is off, and in this particular situation, YAP is free to actually go into the nucleus and upregulate all these different targets, which I showed you in the last slide. So we were interested in looking further into this YAP protein. So YAP, we figured, actually we did this. And we wanted to look first, as we have done for all the other experiments, what is it important for embryonic development? So this has already been shown by another group also in the past, that YAP is important for embryonic development, and we basically showed the same results. So this is control embryos on the left. You can see here different ages: 6 hours post-fertilization, 10, 24, 2 days, 3 days, 4 days, and this is how control embryos. On the other hand, on the right, you can see this YAP morpholino embryos. They look very stunted, they're curved, they're back, there's problem with their CNS also, there is big heart edema, and in general, they do not survive for very long. So YAP, again, it's important for development. So this goes in line, with what we have also shown for HCF1. HCF1 was important for mouse development, it was also important for zebrafish development, and here, HCF1 regulating YAP, and here we have YAP also important for development. So we have, it looks like, there's rinse repeat of these developmental genes which is happening. Now, if we look, during regeneration in the brain, we did a qPCR and looked for all the hippo-YAP pathway components and also for the hippo-YAP transcriptional targets. And some of them we saw were actually getting impacted during regeneration. So we could actually see that some of them were actually slightly upregulated in the target. So for example, caveolin, Lin28, and also, GLI208, which were actually looking, which was significantly higher. So we thought that, we have to now have investigation more into this YAP protein. So we did the labeling and we saw that there was a slight upregulation of YAP protein in general during already at the first day. So this is the first day after injury. So you can see here at the poke site, there's a clear upregulation of YAP, which is available. So now, like I told you, YAP works. How does YAP work? Is basically, when it's nuclear, it's active. When it's cytoplasmic, it's not active. So we looked later, at the 7th day. So this is an immunostaining. This is up is DAPI showing the nucleus. So you can see here, this is actually the poke site, the site of injury, looking like a trough here. So this is, and here this is a YAP labeling. So if you look here in the higher magnification at the poke site, YAP is largely nuclear, whereas here in the other site, below, it's actually cytoplasmic. So indeed, it looks like, that YAP is actually getting nuclear and it's active at this site of injury. So then, we thought, okay, let's look if YAP is also co-localizing, with the BRDU positive cells, that is, they're dividing. So we look here in 4th day, this is a dorsal side. You can see here, this is a YAP labeling here on the top at the poke side. It's also replete with BRDU cells and there's a direct co-localization. This is a dorsal site again here showing YAP labeling, and here, this is a BRDU and there was a localization. So indeed, YAP is also present at the proliferation site. Now we wanted to see that if there would be an impact on brain regeneration on response to YAP inhibition. So we used a drug called as verteporfin. So verteporfin works by sequestering YAP in the cytoplasmic by increasing the expression of 14-3-3 proteins, which basically lead to proteasomal degradation. So here you can see that this is a PCNA is increased in our control upon when there is brain regeneration is happening. But when we are treating these with 0.1 micromolar bodiportfin, it looks like that there is not a substantial increase in PCNA proliferation marker. So now we are in the process of actually looking at this further, and also identifying, through co-IP experiments, if we can actually have a pull down of HCF1 gap and OGT. And also we are trying to look, what is happening to gap in general if you use an OGT inhibitor and so on. So these is all ongoing work. So this brings me, to the summary, what I've described so far. So the conditional loss of HCF1 leads, to increased apoptosis of neurons and glia in mouse brains. At loss also affects the migration of neurons, and it leads to all these different commissural and cortical abnormalities in the mouse brain. On the other hand, in zebrafish, this knockdown of HCF1 leads to reduced survival. There are defects in them. HCF1 is important, looks like, for both brain and retinal regeneration, and the cells, they show a co-localization of HCF1 with BRDU and also the newly generated, progenitor cells, and also the newly generated neurons and glia. Both HCF1 and OGT, their axis appears to play a very important role, and it looks like that HCF1 is probably regulating retinal regeneration via gap. We are in the process of looking if whether this is also happening in the brain. So, we are trying to compare, like I said in the past, through zebrafish and also mouse model systems. We have developed this relevant injury model, and now we are trying to compare the regenerative mechanisms. So a collaborator in ISM-Mohali, Dr. Rajesh Ramachandran, he also has axolotl. So we are also comparing, these mechanisms not only with mice, but also in axolotl. And the hope is that we will be able to find, the molecular and cellular mechanisms which are not only, which are correlative between between different species. So this basically brings me to the end of this project on brain development and regeneration. I would still like to just in 2 slides show you what else is going on in the lab without going into much detail. So as I said, we are comparing fish to mice. So we are also looking at a lot of brain neuroanatomy in the lab. This is in collaboration with a lab in INSERM Paris through a CIPIFRA project. So basically what we do is we do histology of different proteins. So we have chosen WDR proteins. This is a family of nearly 900 proteins in humans, and about 200 in zebrafish. So so Dr. Binaz in INSERM, she already has a library wherein she has done knockouts of several of these WDR proteins, and currently we are collaborating with them to do this histopathological analysis, and we look at the neuroanatomy upon loss of them, and also structural homology mapping. How we landed to WDR proteins is because through a chip-seq analysis I did in my past lab, I found that HCF1, this transcription factor which I showed you in the precedent slides, is actually regulating several of these WDR proteins, and loss of HCF1, it's similar to several of the WDR proteins in terms of brain histology. So we are interested in actually understanding whether HCF1 is basically playing as a transcriptional, a master transcription co-regulator also of these WDR proteins. Lastly, we are also interested in liver regeneration, and comparing it to HCC. We are using different models for ablation of liver cells and comparing them to some transgenic models with fish. In fish, for HCC, we look at gene expression players. We're also looking at different transcription networks. A student is looking at candidate chemotherapeutic drug molecules and looking at their toxicity. We are also doing metabolomic analysis and interspecies comparison. And a student in the lab, Debashruti, she has developed a model for non-alcoholic fatty liver disease in fish. With basically the fish recapitulate the entire paradigm which we see in humans within 2 months. And currently, we are looking at different aspects of this one at transcriptional level and also calorie restriction. So this brings me to the end of my slides. I would like to thank all the students who have actually made all this research possible, and my collaborators, Dr. Rajesh at ISM-Mohali, who assisted us with the retinal regeneration experiments. He's an expert in retinal biology. Dr. Jasprit Dhananjal at IIT, she is the one who has done all the bioinformatic analysis for us. Dr. Weber, who's helping us with the glycosylation studies. Dr. Chetana and Dr. Sridhar at IGIB, who have helped us with the CRISPR-Cas9 knockout and injections, giving us their system. Dr. Padam Shri, who's helping us with the toxicity assays, and also Dr. Binaz and Dr. Stefan, with whom I'm working on the neuroanatomy aspect. Dr. Winship and Dr. Cecile, in whose lab I had done my postdoctoral analysis, and they still helped, with antibodies and different resources. And last but not the least, to thank all the funding agencies, CIPIFRA, DST, DBT, and also IIT for supporting the research. And thank you all for your kind attention. I look forward to the questions.
Thank you, Dr. Shilpi, for the very informative talk. And I must tell you that I have learned a very new dimension from your talk. And before going to the question answer session, I have one question which might be very novice, but I would like to ask in this way only, that whether the difference between the nature of neurogenic niches between this human and zebrafish have some clinical implication. Can we very easily say that zebrafish can tolerate or can handle better the traumatic stress, the traumatic brain situations than human?
Yes, yes. So zebrafish, they are able to actually tolerate traumatic injury, and it's actually been seen that although there is a reduction in zebrafish brain regeneration with aging, something also we are interested in looking at, but still they maintain. I mean, the poke which we actually make inside in the brain hemisphere, it's really big.
Okay.
And then after 14 days, basically it's completely absent. It's not there at all. I mean, it's miraculous how they do it. And like if we do a liver regeneration studies, so basically we take out the liver, we cut a part of the liver and we put it back. We don't even suture it. And 3 weeks later, I see the whole liver has regenerated. So yeah, these organisms, they are incredible.
So really fast in
Yes.
handling the pressure. Okay, so how can we use this kind of study to understand what happens in case of human? Definitively, the links from the genetic makeup, that is helping us from your study only, we can see that. But how in case of clinical situation, how we can compare this kind of situation in terms of human brain traumatic situations? How can we actually map how much the regeneration is with the help of this kind of genetic pattern?
Yeah, so the problem is actually the research in humans, it has not been done. So there have been only, some post-mortem patients in some of the brains they have been seen. And, the data is very sparse. It's really little. So for the moment, all I can say is that, it's really ab initio. What we have seen is that the early response of regeneration is very similar to what one sees, upon low-grade gliomas, when the cancer is starting. So the early events which actually start regeneration, it's very, very similar to the cancer process. And that's all that we have actually been seen in correlation with humans. As far as clinical aspects are concerned, we are actually very far from commenting on that. But yes, what I can say is that since there are a lot of physiological similarities and the genetic equivalence is so high, there's 80% genetic equivalence between the two species. So if anything, we can find, a growth factor or a neurotropic factor, I'm pretty sure that, it has the capacity to go into clinical trials and, to be tested further later on.
Okay. Thank you, Dr. Shilpi. Now I would be going to the question answer session, where I have found out few questions which have come into the chat box, and I would directly be asking those questions to you and get to know the answers from you. So the first question from Dr. Gargi Bakchi is that, have you looked at the role of any GPCRs in the whole process so far?
No, we have never looked at any GPCRs.
Okay. Okay, and do you plan to go for such kind of study in future?
Yes, but as of now, there's no plan actually. I mean, we don't have any of the transgenics for it, but yes, it looks very interesting. I mean, I would be happy. Any clue can tell us, something new about this process. I'm all in, but we haven't looked.
I can see Dr. Bakshi is here. If you have any more question, ma'am, please go to
thank you, Dr. Shilpi, and thanks, Sumisha, ma'am. Thank you for this lovely talk. So your talk actually set me thinking that I wonder if HCF1 is one of the primary genes that are activated at the site of the injury, or would you think that there is a signal which is basically getting transmitted because of which HCF1, YAP, all of these are triggered? So that was the reason for asking about the GPCRs, and GPCRs, you might be knowing that they are very active in the brain and all the regeneration process.
Yes. Yeah, so actually, I don't know what comes first. For the moment, this is all I should say this is all 1 week old data. So, we don't know what comes first. I mean, we are seeing that on first day, I mean, what I've seen is actually YAP come first.
I see.
So the accumulation of HCF1 I've seen is more from fourth day. Also OGT fourth day. It's the YAP. The first thing, also as an MRN target, I saw that YAP is actually coming up first.
Right.
And
Have you done a
so
shorter time point?
So yes, so we have done an hourly thing also with YAP.
I
And
see.
basically, we have seen that already by 12 hour, there was an increase in the levels of YAP. So
Right.
YAP looks like something which is coming up the first, even before HCF1. It's my impression.
So YAP is a co-activator of transcription, isn't it?
Yes, so it looks like maybe YAP and OGT come first and then they bring HCF1, but this I'm just saying this through a very gut feeling. I do not know.
Right. No.
So, but it does look like that.
Okay, thank you. So
Yeah.
because I work in the area of GPCRs and I was very curious to know, whether any GPCR would be involved in the whole process or not.
Yeah. Actually, this field, it's really surprising, but not a lot of molecular and cellular work has been done in brain regeneration.
Right.
So I would not be surprised that, if nobody has actually looked.
Thank you.
Yeah. Yeah. Thank you. You're welcome.
Dr. Minocha, actually, there is a question from our student from Amity University itself, but I think you have already given this answer. Still, I would like to ask you for giving little more light on this model system, which is axolotl, where students are asking whether axolotls, how promising it is to be taken as a model system to study the kind of situation you have made points today.
Yeah, so actually axolotl, I have not worked on axolotl. It's actually my collaborator, Dr. Rajesh Ramachandran, who works with axolotl.
Yes, you have said that
it
has a good
right.
scope.
I'm just hoping I'll get some tissue from him. But actually for brain regeneration per se, axolotl is an excellent model in general for regeneration. And then people have used it for, brain, and most importantly also for limb regeneration, people are using axolotl. Because if you cut a part of the limb, it regrows. But yes, there is high potential with axolotl. It's a very unique model now. The sequence is also available. There are not that many transgenic lines or mutant lines, still available for axolotl because it's a rather new model, and they're really, you can really count the number of labs which work on axolotl in the world. It would be, you can really count them on your fingertips. So the research is very new, but it has great deal of potential. So many correlations have been done on development and regeneration in general with, with axolotl, and even with sharks, people have done these studies. So yeah, the potential is, yeah.
Good.
I can't speak more. Yeah.
So I cannot see any more questions over here. So I would like to thank you from the organizing, on behalf of the organizing team, for such a wonderful and thought-provoking detailed analysis, I must say. And definitively, we would like to hear from you more often, because this kind of science not only gives us the impetus to learn, but also to know many things which basically you will not find in any kind of textbook. So it is indeed a very exciting science, and we would like to hear from you even in person. Amity University Gurgaon itself. Please make sure that while we send you the invitation, you find time for us to give lecture often here. Okay.
Thank you so much.
Yes.
It was a pleasure. Thanks a lot. Thank you so much for the invitation to everybody involved.
Thank you. Thank you, Dr. Minocha. And I would also like to thank my audience over here for their patience. And I would like to thank the organizing team for inviting such eminent speakers between us to have wonderful sessions. And I look forward all to join us in the next session, which is starting at 2:30 sharp. Thank you. Over to you, Dr. Munindra.
Thank you, Dr. Sumishta. So I would again thank Dr. Shilpi for accepting our invitation and taking a time out. Thank you very much for such a nice presentation. For the participants, you have to join back again at 2:30, where we have a session on personalized healthcare, in which Dr. Shipra Chauhan will deliver a talk, which is related to the personalized medicine. So hope to see you all. Thank you.
for Python for bioinformatics and other such courses, and has been editor of a number of journals. She has been awarded ICMR awards for the same. So without delaying much, because we are already a bit late, I would request Dr. Shupra to please share the slides. Please start, ma'am.
You, ma'am. May I stop the video so that the streaming would be better?
Yes, ma'am. Yes, ma'am, you can do so.
Yeah, thank you.
Ma'am, are you able to share the slides?
Yeah, I think this option is coming. Multiple participants. Okay, I can. Yes.
Can you please try moving to another slide so we can just see whether it's working properly?
Is it visible right now?
Yes, it is visible, ma'am.
Next slide also.
Yes, it's
Yes.
moving perfectly fine, ma'am.
So, very good afternoon to all of you, and I would like to thank the organizers to give me this opportunity to present the work and to discuss what are the new areas which are there in the life science sector. So today I got this opportunity to speak about something on personalized healthcare, and right now I have designed this presentation in such a way that we will discuss something about background and introduction. We will see the current trends, what are the major interventions which are taking place right now in the personalized healthcare, and one particular case study where our team has worked in this direction. So before moving forward towards the personalized healthcare, I would like to
Excuse me, ma'am, can we go for a full screen one?
yeah, I was. Is it?
Yes,
is
ma'am,
it
that's
now full
better.
screen?
Yeah,
Okay,
yeah,
okay.
Dr. John, this is better. Thank you.
Thank you. So while we are on this topic, so it's better to understand something from its background. So if and the and as a country of so much tradition and with so much knowledge from ancient time, it is like we should know the personalized health system from the past, which is like what is the historical approach which can be applied here while we are doing the personalized healthcare. So we all know Charak Samhita, and we know that in Ayurveda, there was a personalized system where it is being described that a person, a treatment or a particular prevention of a disease can be handled if we know the prakriti of a particular individual. So we all are aware of Vata, Pitta, and Kapha. These are the three prakritis of humans through which ancient Vads used to prescribe the medicine or the treatment. So it was this approach of personalized healthcare was existing since time immemorial, and we have this written as Charak Samhita, and we know it as Ayurveda also. So we can say that the predictive, the preventive, and the personalized medicine, which is leading from the Ayurvedic concept to human constitution, what we, our Prakriti is all about. So what exactly we are doing here is like we, based on the Prakritis of all the people, we can prevent the disease. We can select the particular drug which is more appropriate for that person. We can customize the whole therapy where we can not only prescribe the drug, we can prescribe the diet, and we can prescribe the lifestyle. We can predict if a person is already ill, how the disease will prognose. That was also there in the personalized system of ancient time. And there was a system based on Prakriti that if, for example, if you are of cough prakriti, there are incidences like there are references that you will be more prone towards the diabetes or cholesterol kind of issues. Similarly, we can, because of this early diagnosis, we can predict the susceptibility. So that was the system which was existing over there in the ancient history of Indian medicine. So now, even with this study, I would just like to provide this view, like it is not just like we are just correlating it without any basis. We are not correlating it without any basis. We can see that there is even scientific reports that where we can correlate the Ayurvedic Prakriti with the genome-wide analysis, which is currently the trend to find out the predictions, to find out the disease, the main disease causing agents. So for example, in this study, they have done the whole the genome-wide analysis, and they were able to correlate PGM1 gene for the genetic basis of the disease. So this is, we can say, yes, the idea of personalized medicine was existing since time immemorial in India, but right now we are revisiting it because we can say that in allopathy or in the modern medicine, the approach was always, firstly, it was the disease-based approach, like where the patients were treated only when the disease was manifested. And the diagnosis was mainly based on the signs and symptoms present by the patient. And treatment was used to be prescribed according to the experience and the knowledge and the intuition of the physician. Then slowly by the interventions of several diagnostic techniques, the disease-based approach, which was basically based on symptoms, was converted into evidence-based approach, where with the help of blood test, with the help of several those markers, physicians then used to make the inferences, and then they used to provide the therapy or the care options to individual patients as guided by these. So now in this era, the allopathic, even the modern medicine, I should say, is also moving towards the personalized healthcare. So what is personalized healthcare? Let's understand that personalized healthcare or personalized medicine is like knowing the or development of patient specific, targeted and tailored treatments provided to the people. So there are two things. The one is personalized medicine for an individual, and the second thing is personalized healthcare. So as you can see that when the personalized medicine is evolving, personalized healthcare is evolving too. So here you can see that by this diagram, I would like to make this point that the evolution of personalized medicine and healthcare is correlated to each other. If you will see vertically, if we are going, if we are progressing towards the personalized medicine, we are also progressing towards the personalized healthcare, which is more of a public approach, because we are moving towards from individual to public side. So here you can see several terms like medicine, personalized medicine, and this is the horizontal progress. Like there was first 3P medicine concept was there, then it came, then 4P medicine was coined, and then the 5P medicine terminology was coined. So here you can see that when we are talking about 3P, so only the predictive, preventive and personalized approach was considered, right? Then came the idea of with the we can say in 2000, when the human genome project was human genome was completed in 2002, at that time, people were more focused towards the participatory terminology also. So while personalized, predictive and preventive was all there, in 4P, they added participatory. So we can say that in personalized, because it takes into account of genetics, proteins, and other profiles of an individual. And because after taking these accounts into these things into account, we can go to we can go for the to indicate what are the best way to optimize the result of a treatment and reduce the side effect or the risk of pathogenesis. So that was the predictive side. And in preventive side, they used to or the physicians try to provide focus on population requiring targets, preventive measures, and not on the disease. So these were the things. Now in participatory, the approach was like we should include the patient also. Okay, so now 4P was came into the picture. After this, another P was added in the evolution of this personalized medicine, and that P was proof based. So the evolution is not stopping till here only, that okay, we are including the patient, we are including the proof also. All the measures, all the predictions which we are taking, we are generating the proof for those that yes, these are ultimately effective for the patient care. So these 5 P's are basically right now driving the personalized healthcare, or we can say the patient-centered healthcare. And because of this, we can say that we are still progressing towards the next personalized healthcare era. So I hope this is pretty much clear with this. Now moving forward, we can see that when we are talking about the evolution of personalized medicine and healthcare, there must be several drivers which are driving this evolution of personalized medicine era. So we can say that there are two things which are driving it. The first one is the digital ecosystem which we have right now, which is more centric towards the individual. Okay, and then we have the research side also, which is like omics and data science evolution development, I should say. So in digital ecosystem, we can see that there are several digital devices, whether it is the smartwatch or the smart applications which are present on mobile phones. Because of these, we can generate digital data, and this digital data is actually providing us the digital insight about an individual. And based on the digital insight, where we can see that okay, these are the changes in the vital signals of a person, we can make the digital decision that you need a medicine or you need a hospitalization. And that is how this digital ecosystem is right now working for individual level also, as well as for public health. So how this individual system is making difference in public health, we will see in the next slide. But here, if we come on the right hand side of the slide, we can see that the omics side. Omics, I think everyone might be really well versed about these terminologies, that the research which is right now ongoing after the evolution or development of cheap I should say next generation sequencing technologies. So the genomics part, the proteomics part, the metabolics part, which is again catering the requirement to understand a person better, whether and as well as the population better. These studies are ongoing, and we have much data about this, and this data is actually the big data. And now with the development of the AI and ML tools, we can make several inferences, or we can make the predictions And based on those predictions, we can ultimately help in making the policies, we can ultimately help in making the preventive tools or methods through which we can go further in the development of next phase of personalized healthcare. So this was all about what are the drivers. And yes, this slide is were focusing on the individual data. And now here we can see that how the individual personalized medicine is actually transforming into the personalized healthcare. So a person who is at the home may provide you data with its smart digital devices. Similarly, the patients who are connected with the clinic, and the clinic is also now well connected with the cloud. So you are not only having the data about an individual, you are having the data about several other individuals also. And because of these things which are present there in a particular population, we have cloud data center which are having the population data, and because of this data we can automate the preliminary results. These preliminary results can be, if you are interested in prediction side, you can go for predictions. If you are interested in diagnostic sites, you can make diagnostic easy, you can identify several markers. So these are the things which are transforming, or this is the ecosystem which you can say is transforming the personalized medicine, means person-centric approach towards the population-centric approach, or you can say the public-centric approach, or the ultimate term, the healthcare. So what is the current trend right now? So every individual is actually enriching the population data. And once we are having a population data, we are actually providing a pattern information. So now we have patterns, and because of these patterns, we can position any individual who is deviating from the pattern. And because this is happening, because we are having the past data, also the present data about that a person, and in future also we will gather the data about that individual. So in individual side, personal data, and in population side, the pattern data. If we are combining these two, we can make better inference, like what will happen, what is the disease probability in future for this particular individual, or in population level, which are the people, how many percentage of people will be having a disease probability. So this is what the current trend is going. So as you can make out from this thing, for the personalized healthcare, it is very much important to have the personal data or the individual data present in a particular population. So we can say that the industrial side industrialists are also right now focusing more towards generating the individual data, as well as they are really interested in having the whole population data, so that they can make the inferences for the better future. So the every healthcare which is present there, the ultimate aim of any healthcare process is to have to provide the better experience to the person or to the individual. So now here, if you can see this, if you can see this news, we can see that even Reliance industry, which is a very big industry in India, they are also very much interested in getting this data. So they have collaborated with several genome like 23andMe startup, which is a genomics based startup, in acquiring the data related to the population, and so that they can come out with some tools later on which may help us in providing the better healthcare. So here in GTU, at School of Applied Science and Technology, our team is trying to work upon this site, and we have selected one case study. So the case study was about the IBD driven colorectal cancer. So here we are trying to work towards coming out towards the betterment of the treatment which is related to IBD and colorectal cancer. So the direction is in that way. And here in this case study, as I told you, the focus is on the towards the treatment side. So we devised the methodology which is based on bioinformatics. And here, just to give the background about this IBD and colorectal cancer nexus, I should say that IBD, which we know, is a inflammatory disease where the immune system of a person actually targets the individual's colon cells, colon lining, and this is termed as the inflammatory bowel disease. And the risk factor of developing CRC, which is colorectal cancer, increases in the patients who are experiencing inflammatory bowel disease. So this is the synergy or the nexus between these two diseases. So we were very much interested in knowing how this nexus is working on the genetic level, and can we identify some treatment which may be helpful in the treatment of these diseases. So yeah, again, just to emphasize on the rise of these, the nexus, the colorectal cancer and IBD. So we can see that there is a rise in the colorectal cancer, which and you can see that it is also showing that it is like in patient group among 15 to 39-year-old, the disease colorectal cancer is like more aggressive, and that is why there should be some intervention. And if we go on the metabolic side of this nexus, which is like from how IBD is converting into CRC, this is a simple study where they explored that the E. coli associated IBD CRC network. So this it was like if the microbiota of your colon is disturbed, okay, so in that case CRC is happening, because the inflammatory induction is ultimately going towards IBD, inflammatory bowel disease, and inflammatory bowel diseases ultimately they're playing role in developing the CRC as the disease progress. And intracellular growth like modulation of cell cycle, DNA repair, and other related events are also contributing in the CRC, which might be induced because of IBD, or it can be directly associated with the increased number of E. coli associated in the microbiota of the colon. So this was more on the physiological side, or we can say on the towards the metabolic side of the disease progression. But when we explored more to find out what are the genetic basis, because we can go towards the personalized health if we can study the genomics or the genetic basis. So here we can see that this is for reference purpose. Here we can see that any disease progression which is in carcinoma, from normal mucosa to carcinoma, several genes are actually playing certain role, like APC here and APC here also. So here you can see that when we did the literature search, we got to know that most common mutated gene in all the colorectal cancer is APC gene, and which is a tumor suppression gene. So by this, so we can say that yes, it is a key player in the disease progression towards the carcinoma. But as we can understand that it is not the case that every time only a single gene is responsible for the disease progression. There might be several other players also. So here, if we can see this example, we can see that in colitis associated colon cancer or in IBD associated colon cancer, some genes are being displayed. Okay, so are these genes really there? Are they the key player, or they are just there? So that kind of study will help us in finding out what are the good interventions for the treatment of this disease. So yes, that was the motivation, that what are the drivers gene of IBD associated colorectal cancer, and can we use some already existing drugs to treat the IBD associated colorectal cancer by targeting those genes which are the key players. So that was the motivation. And here I would like to emphasize, like it is a subtype. The colorectal cancer on which we are focusing was like which is a subtype of colorectal cancer which might have been associated with IBD. So that was the motivation behind this study, and we used the concept of bioinformatics, or the methodology which is there with the help of bioinformatics tool. So the aims and objective were defined as, like, the first project aim was to identify the hub genes or the key genes of inflammatory bowel disease and colorectal cancer disease through in silico method. And the second aim was to study interactions of these genes with some drug which are already known and may be targeted by the those already available drugs. So we have formulated these aims. So just to emphasize, like, we wanted to find, the aim one was to find out the hub genes, or we can say the key players which are driving the disease pathology and the diseases here, in this case the subtype of colorectal cancer which is associated with the IBD. So these are the key players. If they are out, so that means Indian team will not be able to perform as it was supposed to be. So yes, that is why key players are important, or the key genes are important, or the hub genes, because we just don't want it to be focused on only the genes which are present there. We wanted to reach on a junction. That is why we were aiming to get the hub genes. Now the second aim was, like, Fanny de Villiers, who most of the times was able to knock out or to bowl in this direction, that Sachin Tendulkar was out from the game. So similarly, we wanted to find out those drugs which can target the hub gene, and the progression of the disease can be stopped and can provide a better treatment. So that was the second aim of the study. So the methodology which we implemented was in silico approach. All the analysis which took place here was based on the bioinformatics tools. So what we did, we collected the data from for IBD and CRC both, and after the data collection, we were able to identify the regulation the regulation data of differential expression of all the genes which are present there. And with the help of those up and down regulations, we were able to identify the pathways where those genes were actually making the impact. And then we were able to find out the interactions of all these proteins with each other as well as with other genes. And we were then with the help of network analysis, we were finally, with the help of survival plot, we were able to make this inference that yes, these are the what are the key players from all the genes which we have identified, and then based on the key player identification, we did the drug gene interaction studies, where we took 5 drugs and got to know what are the what is the how it is interacting with those drugs. So we can say, if I will go with workflow, like how we collected the data in this in silico approach. So we, in the first step was like the data set of IBD and CRC was downloaded from GEO, which is Gene Expression Omnibus. And the selection criteria for the samples were that they should be from the untreated patients to avoid any co-founding effect of treatment on the gene expression. So with the help of NCBI, with the help of GEO, we were able to download the data set of IBD and the CRC data set. And here there were total 4 data sets for each of these IBD as well as for the for this CRC data set. Now, for differentially gene expressed gene analysis, we used a tool like GEO2R tool, and it actually analyzed the differentially expressed gene between IBD and CRC sample. We used the cutoff of log2 fold change that was like greater than equals to 0.5, and the p-value was less than 0.05. So we use the differentially to analyze the results of this up regulation and down regulation. We use the volcano plot to visualize the genes which are there, and then we identified the common genes which were present in both, which were upregulated or downregulated in both the cases, in IBD also and CRC, or you can say the common one. So that we did with the help of Venn diagram. And then after Venn diagram, we use the for all these common genes, we built a network. So we did the network analysis with the help of string, and we built a protein-protein interaction network. And for the pathway analysis, like all these common genes which are where they are actually playing a role in the pathways, for that we use the David program, and the correlation was, or you can say pathway analysis was done with KEGG as well as GEO tools, online tools or databases which are present. Now, once we were able to perform the PPI networking or protein-protein interaction networking, we identified the key players or the hub genes with the help of Cytoscape. And after identification of hub genes, we we further shortlisted the key player with the help of survival plot. And after the survival plot confirmation, we also tried the hub genes with all the drugs which were into consideration to find out if they can be targeted by these drugs. So this is the entire workflow of bioinformatics thing. Now, if I am talking about results, so there were there were 6 expression gene expression data were there, and they were having all the samples from CRC as well as from IBD. So when we did the these are the volcano plots, and we were able to find out the upregulated genes and downregulated genes for each of these data sets. You can see that this data set was for colorectal cancer. Again, this is also for colorectal cancer, and similarly, the other one was for IBD. Here you can see the upregulated genes which are significant, and these are the downregulated genes, and these genes we did not consider because these were not significant based on the criteria which we took, the cutoff value of log2 FC and the p-value. So now after this, we were able to find out. So the take home message was like we were able to find out the upregulated and downregulated genes here. Now here you can see that for all the cases, we were for 918 genes were coming from this, 265 genes from coming from this, and 688 genes were coming from this. So to find out what are the genes which are common in both the cases, we got to know that in IBD and CRC, 179 genes were overlapping genes which were common in all the data sets. So here you can see the 6 data sets were combined together. So this is the combination of these data set. Similarly, these 3 data set interacted like these 3. So 688 were there, 265 were there, 918 was there, and finally, when we were able to correlate, we were able to find out like 179 genes were present in both the cases. And so ultimately, as you can see here with this thing that once we were able to find out the common gene, we tried to find out how they are interacting and what are the pathways where they are actually playing a role. So we used the KEGG. We used the KEGG database. And we were able to find out several pathways which were present in the disease association. Okay, so after that, we did the survival analysis also. And in the survival analysis, we were able to find out like after the 179, okay, this 179 genes which were common, we were able to find out like 40 genes are also with the help of cytoscape, we were able to identify 40 genes which were present over there. Because cytoscape has 12 different methods through which it can identify the hub gene. So it further came like 40 genes were there. So ultimately, to narrow down the hub genes, what the approach which we used was like we selected only those genes which were present in more than 3 methods or 3 times in the cytoscape topologies. So on that basis, we were able to shortlist only 15 genes. So on those 15 genes, we did the survival plot analysis, and on those in the survival plot analysis, we were able to find 5 genes which were significantly affecting the survival of the patients. So if the simple thing is, if those genes were expressed, the patient's survival rate was very less. So that is how these 5 genes were identified as the hub genes. And like PIMP, CIS-C, CIS-S, CXCL8, CO, CLI, CL1A2. So these were the genes, and based on these genes, we used 5 drugs which are already available, because as you can see that our aim was to identify those drugs which are already available, and we got to know that yes, these like aspirin, metamorphin, and 2 statin drugs like simvastatin and lovastatin, and another one, yeah, sitagliptin was present in the common network where these genes were present. So yes, with the help of this in silico study, we can conclude that this method is quite feasible for identifying the potential therapeutic targets. However, the suggestion is like we should always go for experimental studies to validate this thing, and the approach which we have took is more towards personalized healthcare, because this whole data which we took as the expression data of the patients, it is coming from the public data, and based on that public data, we are trying to provide the better treatment which is actually present right now in the patient and making the disease prognosis. So that's all. That is all from my side. So if there is, I think there is, yeah, there is time. We can, if you have some questions, you may ask. So that's all.
Dr. Munindra.
Costa,
Yes. So while we are still waiting questions from the participants, maybe I got some on my WhatsApp. The first question is, so you have worked with this CRC and the IBD data, and these were more of publicly available data. So what was the sample size? That was a question.
Yeah, the sample size was like, I would suggest, just check. Sample size was different for each and every data set, and altogether it was around 250 individual data.
Okay,
Person.
and connected to this,
Yeah.
so if you think we have to do this kind of study in say in our setup, and it will definitely require a large consortium, because one group cannot have this big patient size available around you. And what are the different legal issues that may arise in doing this kind of experiment?
Yes, sir. Very good question, because that is the challenge which we also faced, because the data set, whatever data set we used, it was not from India. And if we want to perform such studies in India, we are facing the issues like we should have first we should have the collaboration with the hospitals, and hospitals are also like very stringent, like because it is human samples related. So yeah, definitely regulatory and ethical clearance should be there. So that is a challenge. But if we are going to show them these kind of study, that how it is helping us, so which is actually our plan, because we will go to all the hospitals which are available in our vicinity, and we will show them that what kind of this data can make changes, the paradigm shift, and I hope we will be able to do it in our scenarios also.
Ma'am, one question is from my own side. I mean, my curiosity is
Yes.
that if we really predict a drug, or say using your pipeline, we have predicted some key genes or the drug targets, but then the question comes about the validation. So what are the major roadblocks you think in that portion? I mean, can we, based on your prediction, or suppose a predicted drug which may work, so can we directly go for clinical trial or anything like that? Or since it is already in the market as a drug, we actually can skip that?
No, sir. First of all, we need to provide them the experimental, like whether doing cell culture or something like that, where we can actually show that yes, this is actually working. Okay, and on that basis only we can ask for the clinical trials or something like that. So first we right now we are just focusing on the in silico approach. After that we will convert it into the on bench approach, and we will try to find out whether these are actually working by doing cell culture or cell lining experiments. And if the results are going to be fine, then only we will approach for the next step. So these are the things.
One more question we have received. So it is saying, how long do you see it will take for tailor-made marketed formulations to get approval from regulatory agencies?
Sir, I'm not, because I'm also very much in the start initiating phase. So yes, once I will know, I will be able to tell, because seriously, I have no experience as such right now. I'm very novice in this field. I'm just working in silico. I have to convert it into the on bench, and then I will, if I will be successful, then I will go to that side.
Yeah, obviously one session is never enough, because already you have opened up so much in front of the audience. So people are more curious, but as I say, one session is never enough. So we hope to see and hear more from you in future. And I hope your research takes us somewhere, gives us some amicable solution. And maybe if let me just
There
check.
is one more question,
Yeah.
Dr. Kaustav,
Yeah.
regarding TIMP1 gene.
Yes. So you have showed that TIMP1 was a significant gene as the outcome of your study.
Yes.
So what is the physiological significance of TIMP1 in this disease?
Sir, till now I was able to find out this much. I need to study it. What is the physiological effect which TIMP is actually favoring? And right now I don't have this information, but yes, I will study more about it. It is just my preliminary study, because I have to provide like on what I am working towards the best personalized healthcare. I will definitely going to study this.
Sure.
So can I ask one question?
Yes, please.
So do you plan to work on these genes in your next stage of your research? Means your experimental work?
Yes, sir, we are planning. It's basically our team, which is I am from bioinformatics side, and my team is also having one microbiologist and cell culture specialist. So we are trying right now to convert it into that side, where we are actually going to look for these interventions.
Okay. So are there any examples of these personalized medicines which have been approved anywhere else in the world, say in US?
Yes, sir, there are, but right now I don't remember the names. But yes, if you will find, if you will go for this kind of search, there are I don't remember.
I
but
think
if
Roche
APIs,
developed a
yeah.
Roche developed a diagnostic chip on which it was approved for some medicines and for some genes also.
Yes. Yeah, several groups are working, like 1000 Genome is also providing us the pan genome information on variant, and so that we can make the diagnostics more better. So yes, tools are coming, and we hope that we can make some changes. So
And just one more thing I wanted to know. So all these genes that they have already been reported in studies, or these are novel ones?
no, these are already reported. We have just find out are they the key player? So that is the objective. They are there, like they are playing. They see. I showed you one thing, like these are the genes which are present, but are they really playing a key role there in when both the things are there, IBD as well as the CRC? So that was the question which we targeted.
Okay. Thank you.
So we don't see any more question around, but I think we should end with big thanks to our resource person of this afternoon, Dr. Sipra Chauhan. And this was really a helpful session. And as we can find from the chat box, a lot of congratulations are coming out. So we will meet everyone tomorrow in the morning session. But Dr. Ginny, before we end, if you can, if you have anything to say.
Just a small announcement that the feedback link has been shared in the chat box. You just, all the participants can start filling it. It is also available in the WhatsApp group. So please do fill it.
Dr. Chauhan, I would like to thank you for your presentation and wish you all the luck for your team. And I hope to see more of the data which has been technically verified, not only just by in silico approaches, but with lab experimentation, so that we have solid proofs in order to progress for some prognostic approaches. Thank you so much for being here. We again welcome you to Amity for maybe in person for the next
Thank you
time,
so
and
much
we'll
for
having.
giving me this opportunity. Thank you.
Thank you, ma'am.
Thank you. So we remain live for a few minutes so that everybody can fill the feedback. So all are requested to please fill the feedback, and we meet tomorrow again. So tomorrow we have two sessions focusing on nano drug delivery system and neurobiology. So the first session, we have a talk by Dr. Rahul Shukla from NIPER, and he is going to talk about drug nanocrystals. And in second session, we have Dr. Nitin Chitranshi from Australia, and he will speak on eyes on neurodegenerative diseases. So these are two exciting talks which are coming up. So do join us for the sessions tomorrow. Thank you. Please fill your feedback forms.